is Mazyar Fallah, who is joining us from Guelph. Maz got an undergraduate degree at Johns Hopkins. Then he did a master's and PhD with Charlie Gross at Princeton University. That's where I met Maz. I was an undergraduate in the lab when he was a grad student, and then I followed his footsteps and did a PhD in the same lab as Maz. So I've known him for a long time. After that, Maz did a postdoc with John Reynolds at the Salk Institute. Then he was hired at York University in a research position in the School of Kinesiology and Health Science. And he gradually took on more administrative roles in addition to his research, became Associate Dean of Research at York. And then 2 years ago, almost exactly, was hired as Dean of Biological Science at Guelph, where he is now. And throughout his research career, his research has focused on visual perception, eye movements, attention, and how visual objects are bound together from lots of different sources of sensory information, and how this impacts our eye movement behavior and attention. His talk today will be continuing to talk about oculomotor behavior and perception. So take it away, Maz.
Thank you, Dylan. Oh, this is great. Thanks for having me. And as Dylan said, we've known each other for a very, very long time. Today, I'm going to talk to you about oculomotor modeling and what it tells us about cortical processing. And I just want to check, make sure I'm going to do this a lot. So hopefully the people online can see me do this when I point instead of trying to use the little cursor on the screen, because that's a lot more natural. Oops. So the oculomotor system, as depicted here, there's a lot of different nodes, cortical and subcortical, that control it. But what's clear from this picture is that we're not actually including visual processing areas in it. We know for motor actions, we need our other senses to guide those actions, and that's considered sensory motor integration. But for the oculomotor system, especially for saccades, those rapid jumps you make from one place to the other, the argument has always been that at most what you have in the oculomotor system is a map of priority spaces. And so different locations are prioritized based on the objects that are there. And that's what you use to create motor plans to cause the eyes to move. And that's where you get competition between the motor plans in either a vector average method or a winner take all method. And I'll get into those later in a bit more detail when I talk about the research. But that's why this map doesn't really include visual processing, because you aren't supposed to care about the visual processing. You're supposed to care about the value that the object is that gets fed into that last priority map, regardless of what features or what type of object it is. And obviously, if that were the case, I wouldn't really be here talking today. So cortical areas aren't included directly in the network. Why do we study the oculomotor system? Just in case people are more familiar with other systems. Eye trackers are everywhere. Your phone's got one. Your laptops have them. Anything that's a camera these days can track your eyes. There are insights into thoughts. If I stare at something long enough, you have an urge to stare at where I'm looking, right? If I look interested when I'm talking to you, you know I'm interested. If I look bored, you know I'm bored. We use eyes and gaze to understand what other people are thinking. It's a simpler movement system than a lot of the other movement systems because it's a 2D projection. Yes, the eyeball is a three-dimensional object and it rotates in the orbit, but we actually encode the motor plans as 2D vectors, as if we're moving the image along a 2D retina, even though the retina is also curved. And the saccades have been considered ballistic movements. Ballistic, fire and forget, like a cannonball. You plan it, you fire it off, and it lands, and you're good to go. But are they really, truly that ballistic? We can take a look at saccade trajectories, and there's old research on this. The first one, obviously, is that if you fixate here and there's a target, you make a saccade or an eye movement to it, you go pretty straight to it. But if, as you're about to make that movement, I turn on a red dot near it, now your trajectory actually curves towards the red dot, and your endpoint falls in between the two. You're no longer going directly to it. And we can play with that. If we leave that dot on all the time during a block, you know it's a distractor, you know it's irrelevant, you're supposed to be looking at the blue thing. Well, you'd think you'd go back to straight, but you don't. You actually shift away in the other direction. So your movement is curved away and your endpoint is shifted away from it. And that's the sign of inhibiting the distractor. And so the way to think about this is when this comes on, there's a hill of positive activity. That excitation in motor plans in the oculomotor system gets combined with your plan to the target, and you get a curvature that falls in between. And if you have a long enough time, you can inhibit it, and then it inhibits below the baseline rate. And so now you've got an unequal weighting with inhibition here and neutral weighting everywhere else, and that shifts you in the other direction. We can take measurements of this out of the lab doing the research. So you've got a target and a distractor, and there's a number of metrics. The two I'm going to primarily talk about today are saccade curvature, which is taking a look at the starting point and the end point of the saccade and summing the area under the curve. And endpoint deviation, which is the difference between the vector you're supposed to be using to go to the target and the vector that you actually create, and that's in angular coordinates. So what mediates this excitation or curvature towards versus inhibition or curvature away? The answer is time, how long you have to process the distractor. Now, how do we get there? Simple task. You fixate, fixed spot goes off, and a target appears. Red dot is a depiction of your eye location. Your only task is to make a saccade to that target. If after a random period of time, we put on a distractor, pop it on randomly, we can get curvatures towards or away based on that timing. We're going to see that we use two distractors, and isolumina color grading is depicted here, or luminance grading. So we're looking at distractor processing time, which is defined as the time between the distractor onset and the saccade onset. So the more time you have to process the distractor, the more effect it can have on that saccade to the target. And it makes sense. If the distractor comes on right before saccade onset, a millisecond before, there is no time for it to be processed through the visual system, through sensory motor integration, and affect your motor plans and your motor output. But as you give it more time, as there's more time between the onset of the distractor and the onset of the saccade, now it's being processed by the brain, and it can start having effects on the saccade itself. And what we have here are all of the trials that were split up, all the trials that were towards the distractor in terms of endpoints were analyzed and summed together, and all the trials where the endpoint was away from the distractor summed together. What we've done is taken every single saccade trial, measured the distractor processing time for that saccade trial, binned them in 20 millisecond bins, and then measured the curvature. And we plot the average curvature for every bin of distractor processing time. So we have thousands of trials per participant and multiple participants to be able to give us enough data that we can then fit it with a Gaussian for each of these, whether the curvature was towards or away from the distractor. So we're getting what's called chronometric modeling of the curvature response based on the timing of the distractor versus the saccade onset. What comes out of this is something really nice. We can see that when there is a short period of time between the distractor onset and the saccade onset, so the distractor processing time is short, it's attention grabbing, it's attentional capture, and that attentional capture drags the curvature of the saccade to the target towards the distractor. You want to look at the distractor while you're trying to go to the target, so you curve towards it. If you give it more time, then you overcome that attentional capture and you're able to inhibit the distractor. And as you inhibit the distractor, that inhibition also has its own time course. So the inhibition builds up over time, reaches a maximum, and then drops off over time. And it sort of makes sense. If something's in the room that you wanted to inhibit or from distracting you for a time, it doesn't last forever. It might be important later. And so the distraction or the inhibition drops off over time. Now, nobody had actually determined this in terms of time course. The prior studies that looked at distractor inhibition had the distractor on the whole time. The distractor was always on before the target, so you would see it. You'd miss out on the attentional capture. You would know it's a distractor and you would inhibit it before the target ever happened. And they found curvature away, but that curvature away doesn't drop. It stays on and on and on because the distractor is in the same place from trial to trial. So what we found here is a new form of distractor inhibition, which is rapid inhibition. Something appears on, just like in the attentional system, it has attentional capture, then goes into inhibition of return. We're finding the same correlates in terms of the eye movements. But what was even more interesting is they look similar. In fact, if you take these normalized functions and you overlay them so that the peaks line up, the two curve fits are basically the same, 19.6, 20.8 for those peak points. The shapes are very, very similar. And so what we see is that the effect of the distractor, whether it is an attractor or a repulsion from inhibition, it's the same circuit. It's the same function. The only difference is the sign of the effect. So the circuit operates the same for excitation and inhibition, but based on the timing of the distractor processing time, it flips in sign. How it does that, we don't know yet, but we've got neurophysiological recorded data from FEF doing just this task that we're going to try to understand exactly what's happening there. Now, I can give you a putative model for it, and this is based off a lot of work of McKeek and Keller that looked at most of these timings doing recordings in the colliculus. And this is complex. I'm going to just minimize it to one thing. When you make a saccade, the neuron that encodes that saccade has a critical epoch right before the saccade onset of about 30 milliseconds. If another motor plan feeds into this time period, that's when you get changes in the saccade trajectory metrics. So we've got our target alone, our critical epoch. If the distractor appears with very little distractor processing time, we have the excitation or the onset burst and the response of those neurons to the distractor, and we get a positive vector towards the distractor. But if there's more distractor processing time, the onset is here, that excitatory burst happens, and then we get a refractory inhibition that occurs after it. And this inhibition during the epoch causes the curvature away from the target. This is hypothetical. This is what we're looking for in the FEF recordings. Now, if you recall, we use both luminance contrast and color contrast as our distractors. And in doing that, we could split the data, not worrying about whether it was excitatory or inhibitory. We just collapsed all the data, rectified, so everything is in the same direction. And take a look for a difference between the stimuli. And this is, again, where something really interesting pops out. The luminance contrast has a peak effect at 141 milliseconds. The color contrast has a peak effect on saccade curvatures at 151 milliseconds. They're 10 milliseconds apart. So this oculomotor modeling, if we know what the peak of the response is, we know what type of stimulus we used. It differentiates between a luminance contrast grading and a color contrast grading. That is very different than what we would normally expect for a distractor onset. Distractor onset that fed from the retina to the colliculus doesn't really care about these features. You'd expect the timing to be the same. This timing difference is the timing difference you get between area V1 and V2 and area V4. So we're seeing a difference in the effects on causing the saccade to curve based on what stage of visual processing along the ventral stream the stimulus is primarily defined as. So to start off, we can use the spatiotemporal profile or chronometric modeling to show target selection. We get excitation and inhibition, but they're equivalent processes with a sign flip, and the color distractors delay the trajectory effects by 10 milliseconds, consistent with V4 over V2, even though a generic onset response would be the same. And there we go. And therefore, this is the first evidence that we're going to show that cortical visual processing is integrated into saccade curvatures. It's not just this final map that takes all the ventral stream processing to the end that matters. It's getting information at what stage along the ventral stream is the peak activity for the type of stimulus that's there. And that's a bold statement now, but I'm going to spend the next couple of studies showing you that that statement is true. So we're doing this non-invasively as a way to understand the underlying neuronal circuitry. So we've done it for color and luminance contrast gratings. Let's take a look at something a lot more complex. Let's look down the ventral stream into IT cortex, where we have late stage object representations, and we created these new novel complex objects. So it's a 2x2 grid, but has a whole series of line segments. And some of them are on and some of them are off. And from the first stimulus to the second, there is a 1 segment difference. So this segment is different between these two. And from 2 to 3, now this segment is different between 3 and 2, but we've got 2 segments different between 1 and 3. As we go along these stimuli, none of these look exactly like letters. We removed anything that looks like a letter. And so this is the stimulus set that was used. But what we have here is a way to actually measure the similarity between any two of these complex objects objectively. So stimulus 3 and 4 have an objective similarity of 1. There's only 1 line segment different between them. Stimulus 2 and 5 have 3 line segments different between them. Objective similarity of 3, 1 and 6 have an objective similarity of 5. And what we're looking at with the complex objects is to see if the saccade trajectories reflect the similarity between a target and a distractor. Can that saccade trajectory, does it encode the difference between target and distractors? Does it reflect the target distractor competition in the visual area that would distinguish them? So these stimuli are specifically targeting areas TE and TEO. And the task, because we have to have similar stimuli, we had to go from a distractor-only task that was task irrelevant to a search task. Participants fixate. One of the stimuli appears as your target. You have as much time as you want to memorize it, then you press a button. You fixate, then the 2 stimuli come on with a random period of time between them. Equally possible for the target to appear first or the distractor to appear first, but we're only going to analyze the trials where the target appears first, the distractor appears later, so we can measure distractor processing time between the distractor onset and the saccade onset. So here's some actual eye movement trajectories, and we've color-coded them by the distractor processing time. And you can see that there's a bit of a gradient where the shorter distractor processing times are showing curvatures in one direction, and the later distractor processing times are showing curvatures in the other direction. We can plot this out. And again, we've color-coded them by the times, but we've gone away from binning. We're using a bit more of a better method for the chronometric modeling. And that's why we're not seeing them in bins. But what we can see here is that in this early stage, around 240 milliseconds or lower, we're calling it the active decision-making stage. This is when you're still looking at both the target and distractor and trying to decide through your active discrimination which one is the target that you're trying to find. After a certain point, you've made that decision. The decision-making is over. That's when you can start inhibiting it. You cannot start inhibiting the distractor until you're certain it is the distractor. Pretty simple rule there. And so in the early phases, we've got curvature towards the distractor. And then after decision-making is over, distractor inhibition starts, and you've got curvature away from the distractor for saccade curvatures. For endpoint deviations, we see the endpoint is shifted towards the distractor early, but once it's over, it's actually quite flat. So we're getting curvature, we're getting endpoint deviations towards the distractor, but we're not seeing the effects of distractor inhibition on the endpoint deviations. So let's take a look at the main question. What happens with similarity? We split up those graphs, and this is saccade curvature and endpoint deviation again, by similarity levels, and in 3 different differences. a small difference OS1, a medium difference OS3, and a large difference OS5. When you split this up, let's take a look at the inhibition period. So these black lines show where there was a significant difference between the three conditions in our inferential statistics. And what we see for Saccade curvature is that curvature starts earlier for OS5 than OS3 or OS1. We also see that OS5 shows more curvature away than OS3 and OS1. What this is saying very simply is that the greater the difference between the target and distractor perceptually, the sooner and greater the inhibition happens. So it occurs earlier and it's stronger. And so the curvature effects we're seeing are a direct reflection of the perceptual discriminability between those objects, which can't be handled by the visual motor neurons in the colliculus or in FEF. It has to come from the visual processing in the area that discriminates them, TE or TEO. We find the same thing happens for endpoint deviation. You know how I said there was no effect? It's actually quite interesting in that OS3 and 5 are actually have the endpoint shifted away from the distractor. OS1 is actually still shifted towards the distractor in that period. So what this is saying is for endpoint deviation, if it's really similar, it can't be inhibited and it continues to drive a competitive eye movement. But for greater differences, the distractor is inhibited and it's separated. Why are these different? This is a vector representation that we suggest is a competition between motor plans in the oculomotor areas. And this is a real-time online input from the visual areas that are processing the features and are showing target distractor competition in the visual areas. And that's why we're seeing different patterns between the two, and that'll be replicated in later results. So when you search for a target, Saccade trajectories reflect how similar the other objects are. When you're actively discriminating between the items on the screen, the distractor is positively weighted by the oculomotor system, but when you complete that discrimination, decision-making is over, then you can inhibit it. The target distractor competition in visual areas feeds into the competition between the Saccade motor plans, and these late-stage visual processing areas like TE or TEO have to provide weighting for the competing Saccade plans based on similarity that only they can compute. Now I'm talking about features that are processed in the ventral stream, luminance gratings, contrast gratings, and these complex objects. And the ventral stream hasn't really been linked to the eye movement system, but the dorsal stream has. And in the dorsal stream, it's been linked there because of motion. Motion processing area MT and MST are linked to the oculomotor system. They help you do everything from pursuing with your gaze a moving object to intercepting with your gaze something that's moving. So we know that the motion system is integrated in a dynamic network with the oculomotor system. And the question on this study was to see if we could use motion processing to affect the Saccade trajectories. So the hypothesis is that if the motion moves towards, well, that one's away. If the motion moves away from the vector to the target, it would actually drag the curvature and the endpoint over with it. And if the motion moves towards it, it would push it away. And that would be on top of the effects we saw for the distractors from the ventral stream. So we expected at least a partially independent modulation of what we saw before. We all see is two speeds, fast and slow. There is an argument that there are two different speed channels in the motion processing system, slow and fast, and that the faster you're moving, the more motion strength is recorded in those circuits. And so the argument is that a higher motion strength stimulus, the faster speed, should show greater effects on the curvatures and endpoints than the slower motion. And we had a static as a comparison. So when we take a look at motion direction, towards is green, away is yellow. We're taking a look at this curvature period here, and there is no difference between towards and away in terms of the peak amount of curvature, or on that side, the peak points in time. But what we did find was a significant difference in the onset of curvature between static and either towards or away, and in the peak activity of static versus towards and away. So what we're saying is the motion direction did not matter for the Saccade trajectories. But if the stimulus was moving, the effects on the trajectories was delayed. Sort of that same difference we saw between luminance gratings and color gratings, we're seeing it again between static and moving. That's not there. There we go. We can take a look at motion speed. And once again, there's no difference between fast and slow. But again, we see a difference between static and fast and slow that are the same. So we can combine them. So put both speeds, both directions together as here's a motion stimulus and here's a static stimulus. And not surprisingly, because it was significant on the last two figures when we only used the data split up in two different ways, you put it all together, it's still very significant. So what we're finding here is that when there is movement in the distractor's aperture, it delays the effects of the distractor on the Saccade trajectories. That's crazy. The reason why I want to say that's crazy is first frame. The first frame of a motion stimulus is a static frame. In the static stimulus, it never changes. So we put up that first frame, and that first frame in the static stimulus affects the Saccade curvatures sooner than if you change that frame and you change it again and you change it again to give you a motion stimulus. The first frame is the same for motion or non-motion. And you would expect that if the static gives you an earlier response, that response would be feeding through the system, even if that stimulus then moves afterwards. But what we're finding is that it doesn't matter for the first frame. If the stimulus starts moving, the effects delay until the motion is processed. So we're going to go out on a bit of a limb here and suggest that the processing time of the primary feature defining the distractor determines when it affects the Saccade programming. What this is saying is that the visual system is not a linear line system feeding into the oculomotor system. It's not about the earliest responses in the visual processing hierarchy. What actually matters is the area that distinguishes the features of that distractor. And so it's dynamically connecting to the area that gives you the peak activity. That first frame will activate V1 and V2, but the motion will activate MT. And so MT activation is later than V1 and V2 activation. Therefore, the response is delayed by the difference in time between V1 and MT. And that's what we're seeing here. And that is hard to believe. I mean, the data's here, you should believe it. I mean, in terms of circuits, because we think that it's a labeled line. The idea was always vision processes it, you get these little bumps of activity and off it goes. And it suggested it always flows through the whole system. So the earliest activity drives it and the last activity drives it. We're saying no, it's dynamically being driven by the area of peak activity along the visual processing system. So with this as a theory, we need to test this. Can we show through this modeling that we can find latency times across all the areas in the ventral stream? Well, I've shown you oriented gratings, color gratings, and complex objects, and that's what we're going to do. So we can take a look at timings from primate neurophysiology between V1, V2, V4, TE, and TEO, and get those timings. And we're going to do the same thing with the stimuli I just showed you across these studies: luminance, color, motion, complex objects. And we expect that this oculomotor modeling would show up the earliest latency responses for luminance, then color, then motion, then complex, as we're going through the system. So here's the motion data again. As we've gone through these studies, we've actually improved on modeling and the stats and the inferentials and everything that's going along with it. So we're getting better. And so we reprocessed all the data with the new methods. And we can get the onset latencies using a Gaussian kernel of 10 milliseconds and sliding t-tests. And what we find is that it takes 81 milliseconds of distractor processing time to start having an effect, driving the curvature towards the distractor for a motion stimulus. The peak of the activity occurs at 111 milliseconds. So 81 should be the time that we're looking for the latencies in areas that process motion, and 111 is the peak activity in that area. We do this for our 4 different types of stimuli, and we get a clear pattern of results. The luminance grading is the shortest at 77 milliseconds, followed by our motion processing latency at 81, followed by color processing at 99, and our complex objects at 135. And everything is significantly different from everything else except luminance gratings and the motion stimulus. Those are 77, 81, they did not come out as significant. But we're seeing this pattern that comes out here that matches what we would expect for visual processing. We can do this for the peak latencies as well to get at that idea of the peak activity in the visual system is driving the effects of the curvature. And if we do this again, now we see that at 100 milliseconds, we've got our luminance grading. 111 and 112 is motion and color, and then our complex objects at 155. These are all longer because the latency is when the area starts activating, and this is the peak of activity in those areas that we're modeling. So if we take a look at all that data, it's pretty simple to see. We take those numbers, we plot them up here with the stimuli, and we look at what was found in the monkeys. So our luminance grading, 77 milliseconds, falling in around V1, V2. Color at 99 is also a little longer, but again, in line with what we'd expect for V4. Complex objects fall in there. Motion falls quite close. It's either V1 or V2. So a couple of take-homes. We are seeing a progression in processing along the ventral stream that matches what we see when we do neurophysiological recordings. All of these times are slightly longer than all of these times, but these were in the monkey, these are in the human. So that may be the reason. It could also be part of the methods because we only have a certain level of resolution to be able to see off of the oculomotor modeling. And maybe at the neuronal level, it's easier with firing rates to have better resolution for the leading edge of the latencies. You know, this really is about the population. So we need enough neurons firing at the same time to cause that shift. And it should be a little later than we see here. And it suggests that the motion stimulus is actually occurring with motion cells that are in V1 or V2. We know there's motion responsive cells in V1 and V2 rather than MT, which is why the onset latency is actually similar to the V1 latency. All right. So cortical visual feature processing affects Saccade vectors. The onset latencies reflect latencies along the visual processing streams. Peak activity reflects the visual area with the greatest activation for that feature. And this modeling allows us to estimate latencies of visual processing along and across the 2 visual streams. We can do this non-invasively. So now it can be used to ask the questions about when's the latency of any type of complex stimulus. When is the time that the area starts firing to it, and when is the peak activation? So when you have a stimulus that affects a lot of the visual system, but you want to know where is it primarily being driven by, we'll get a measure of that through this system. You can also take a look at it when you want to modulate visual processing using attention, inhibition, habituation, pharmacological modulations in disease cases. Anything that is going to affect visual processing, you should be able to assess its effects on latencies and peak responses through this method non-invasively and very easily. With all that in place, I must say all of those studies were done with the distractor at 45 degrees angular distance from the target. It was always a set distance. And so what we needed to do then was see how those responses, how those results generalize across space. Because in the real world, not everything is always 45 degrees apart. So it's the same task, target, memorize it, 2 items appear. And then we actually varied the distance between the two items, but we didn't vary. We varied the distance between the two items, and we varied the objective similarity between the two items. So it occur anywhere around the circle, but we code everything relative to the target as +1 to 5 or -1 to 5 clockwise and counterclockwise, just to collapse the data. There's, as I said, a number of metrics that we can use. 3 are involved with curvatures, but again, we're only going to focus on some curvature. 2 of them are involved with the vector representation for that saccade, initial angle and endpoint deviation. Initial angle is when you fire off the first 20% of the movement. What is that angle? And endpoint deviation is the endpoint relative to the target. So we're going to look at some of curvature and initial and endpoint deviation separately. One, initial angle really is a type of vector averaging of saccades to the target and distractor. Endpoint deviation is very similar, but it allows for error correction during the curved flight. And so endpoint deviation can be slightly more back on target than initial angle. And we wanted to see what the effects were for the curvature in flight on the saccade vector metrics as well. So if we look at angular distance for the short times, oh, sorry, I forgot to describe this. So this is the chronometric processing, short distractor processing times, long distractor processing times. The solid lines are where there was, in that time frame, a significant curvature or endpoint or metric towards the distractor. The dashed lines are where there was a significant metric away from the distractor, so distractor inhibition. And these boxes in the middle are the transition zones. In these transition zones, there's no significance one way or the other. And it could be that you have curvature towards, flat neutral, and then curvature away. But in fact, what you have is a mix. So if you look at any point in here, you get a Gaussian distribution that shifts across the transition zone. So it says on a trial by trial basis, the inherent noise in the system has a temporal variability. And what we did then was remove the temporal variability so we don't mix some curvatures away with curvature towards by having a hard set time. And we have this. There's actually some interesting results that come out of that in terms of how these metrics group and how the processes are ordered in the oculomotor system that I'm not going to cover. But here are the average trajectories across 5 angular distances. And you can see that as the angular distance gets further out, the curvature gets larger. But as the objective similarity goes from 1 to 4, it's actually a much smaller range. We can look at the long times when the distractor is inhibited. And again, we can see a general pattern that as the angular distance gets further, we get more curvature out, and we get a little more variability for objective similarity. So we can take these and now do our standard modeling off of it. I'm not going to show you the modeling, but what I am going to do is take the metrics of the modeling at each of the angular distances and plot them. And the question is to see what is the effect of space on these saccade metrics. So on the target distractor competition, the space or the distance between the target and the distractor matters. If we're looking at curvature, when they're really close at angular distance 1, there's not much curvature. So they're 22.5 degrees apart, but at that distance, the saccade tends to be straight. So the endpoint will be deviated between the two, but you're getting a straight saccade between the two. And that reflects vector averaging. But as the angular distance gets larger, the curvature starts increasing towards the distractor, but you reach an asymptote. There's a limit or a max function in this circuit. And so it's basically saying that at 67.5 degrees or larger, there is a limit to the effect of the distractor on dragging the curvature towards it. Anything from 67 degrees out to as far as we tested has the same weight in dragging that curvature outwards. If we take a look at the endpoint deviation then for the vectors, not the trajectory curvatures, we see this different pattern. As I said, at angular distance 1, the saccade vector actually falls in between the two. As you move out with more distance, the saccade vector actually moves closer towards the target. In the vector average model, you would say that if you had two vectors, 0 degrees and 45 degrees, you should fall in between at 22.5. So the further we pull out the distractor, the further out you would expect the endpoint deviation to be. But in fact, that's not what happens. What you get is a decrease based on distance. And what this is saying then is that there's a weight to the distractor compared to the target that follows this pattern, that follows an exponential decay. As you get further out, it has less interaction with the target on the endpoint deviation in that weighted average between the target and distractor vectors. So we've got different effects between these two. This one is showing an effect that should be related to the local circuits in the area of question. So we're looking at complex objects in TE and TEO. We know the size of the receptive fields. We know the mapping in these areas at least in the primate neurophysiology. This suggests that 67.5 degrees or less is the extent that local interneurons can cross that cortical area. So we expect that this is actually a measure of local interneuron circuitry, at which point it goes beyond it, which means if we replicate this with an earlier feature like luminance gratings in V1 and V2, where the local interneurons have the same length but cross a larger portion of the cortical magnification, we'd expect this number to change by area. But this is actually vector competition between the target and distractor in oculomotor areas and not visual areas, which is why we're seeing this exponential decay over time. The further apart they are, the less the effect is. And again, it asymptotes out after 67.5, which says that in the oculomotor areas, the competition also has a limited range from the local circuitry, at which point the distractor has no effect on the endpoints. It's not vector averaged anymore, and that has to be a local circuit. We can look at the long distractor processing times when the distractor is inhibited. And here we find something very different again, and very interesting. What we're finding are spatial surround suppression. Spatial surround suppression suggests, and it comes out of attentional models, such as John Sotos' selective tuning model. It suggests that when you have your attention focused on a point, anything within that attentional spotlight is advantaged. But then right outside the spotlight, you get an inhibitory zone, so you can help focus on the spotlight in terms of center surround interactions in the visual system. So you get this increasing inhibition that then drops off, and at some point then it's neutral further on. And that's been found behaviorally in a number of areas for very simple features. When they're looking at it spatially, they were looking at a variety of color, motion, or simple shapes. We're finding that result here for these complex objects on the trajectory information. And so that suggests that the spatial surround suppression is either repeated in motor areas and sensory motor areas the same way it works in sensory areas with attention, or, as we have been positing, curvature is being fed dynamically real-time information from the visual area that has peak activation for that stimulus. So we're seeing the spatial surround suppression that we know would be in TE and TEO reflected in the amount of inhibition that's happening to the distractor that we are measuring in terms of our Saccade curvatures. Same thing's happening for endpoint deviations. We're still seeing spatial surround suppression for endpoint deviations because this is all about location and space. It doesn't care what the object is. It's a spatial code, and that spatial code is replicated both in the vector and in the curvature weightings. What the key then is what happens when we take a look at similarity. So when we're looking at similarity, there's this thing called feature-based surround suppression. It happens in feature space for simple features, which says that if you have an orientation, nearby orientations are advantaged. As you get a little further off, you suppress them, so you get better discrimination of those small orientation differences. And then once you get far enough along, now there's no longer any suppression. So it helps improve the selectivity and resolution of simple features. It's been shown for orientation, it's been shown for color, it's been shown for direction of motion. It's never been shown for complex objects till now. So we're finding that the type of coding and type of discrimination circuit that occurs in early visual areas for simple features is likely replicated in late stage ventral stream processing in area IT. So when you're no longer coding simple features, but actually encoding objects themselves, we see the same circuit working again. And so this is surround suppression, but not spatial, not feature-based, but object space-based surround suppression. And this, as far as we know, the first evidence of object space surround suppression existing. So conclusions: active competition is limited to short distances, likely due to short-range neuronal connections in the local area, and gives us an idea for the underlying neuronal circuitry that is involved in these target distractor competitions. When the competition is complete and we can inhibit the distractor, now we're seeing spatial surround suppressions out of that inhibition, strongest at the middle distances and predicted by attention models, which then again suggest that the curvatures in these cases are being driven by active dynamic connections to visual processing areas. And the similarity effects during the active competition, and this is important, right? It's active. You haven't decided which one is the target yet, so you're processing both. And that means the areas like TE and TEO are actively trying to discriminate the two. That's when object space surrounds and things like selective tuning would be at most effect in non-spatial locations like object space. We're seeing the first evidence of object space surround suppression here. And this suggests that our understanding of representations in IT cortex, as with face space says that you define faces along different axes in face space between facial features, and that's the code for faces. That type of idea of object coding in cortical areas like IT generalizes to all sorts of objects you have no experience with, like these novel objects, because we're seeing object space surround suppression. The same type of object space coding should be generalized across IT. And with that, I'd like to thank the grad students that worked on this, Devin and Caroline, a number of undergrads who worked there, including a MITAC's Global Summer Research Intern, funding from NSERC and from CIHR, and the list of the papers that have been discussed in this talk, 4 that are published, 2 that are in review, and we received reviews and are revising for resubmission to them, responding to the reviews. And with that, I'm happy to take questions. And I'm just on time.